we're going to go again like we normally do, or regularly end up doing, from very, very large and very, very far to very, very small and very, very close.
Very, very small now, yep.
With our next story out of the University of Zurich, which is headlined, AI meets CRISPR for precise gene editing. And the byline is a research team headed by the University of Zurich has developed a powerful new method to precisely edit DNA by combining cutting-edge genetic engineering with artificial intelligence, and the paper was recently published in Nature Biotechnology.
Yeah.
So we've talked about CRISPR before. I think most viewers who have come across this video will be relatively familiar with what CRISPR is, so maybe we won't do a deep dive on the basics.
Yeah.
But we can touch on what is the insight or improvement here that has allowed them to increase that precision of what has been a revolutionary discovery with the underlying CRISPR work that has already been done.
Yes. Yeah. So CRISPR, it's kind of an old technology now.
Right.
It's almost 10 years old. And okay, CRISPR. Okay, you guys happy? You know, it's I've been getting all this shit.
That's true.
From these TikTok comments.
It is spelled, with CRISP-R. It's an acronym.
Yeah, it's an acronym. It's an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's not a real word. I can say it however I want. But I'm gonna say CRISPR now.
It's
I got bullied into it.
The peer pressure worked.
Yeah, the peer pressure works, dude. So CRISPR, it's pretty old technology now, almost 10 years old, maybe a little bit more. Basic idea: you get to cut DNA wherever you want, in like the spot that you want. Before, you could only cut DNA if you looked at certain base pairs and then recognize those. With like restriction enzymes. Now you've got a custom restriction enzyme that you can be like, okay, I want you to look for ATCGGA, and then wherever that is, find it and cut it. That's what CRISPR can do. CRISPR only cuts. And once it cuts, other stuff comes in to repair. Okay? And so far, it's just been a sort of cut and hope. Okay? You cut it at the spot. And you hope that whatever machinery is in the cell already is going to come and repair this DNA. Right. And there's not going to be anything weird that happens. Of course, that's not always going to be the case. Of course, you're going to get lucky. Most of the time, you're going to get lucky if it's a healthy cell. But in certain situations, it's not going to go well. Right? There's going to be these repair mechanisms. What they're going to do is sometimes delete a nucleotide or two, and then your whole gene is completely wrecked. You might be working with something called like non-proliferating cells, so cells that don't actually cell divide, like your neurons. Skin cells cell divide. Most of the cells in your body, muscle cells obviously cell divide. Your neurons do not cell divide, so they don't have half the machinery necessary for DNA repair that a dividing cell would have. Dividing cells, they're constantly like replicating DNA, checking to make sure the DNA is correct and all this stuff. But in your neurons, it's like, you don't need that. Right. This neuron is going to live until you die, and it's not going to make any more. So for those cells, if we want to use CRISPR and these new genetic technologies, we're going to need a more robust way to control for the repair after the cut.
No more cut and hope.
Yeah, no more cut and hope. We want to cut and then engineer. Right. The cutting was an engineer. Before it was like the cutting was a hope too. And then we upgraded. It's like, okay, now we can engineer the cut. Now this is like sort of going towards engineering the repair. Okay? They made this AI called Pythia.
Okay.
I hope I'm pronouncing that right. Okay, whatever. It's named after the oracle at the Temple of Apollo in Delphi. Yep. Have you seen 300?
Yes.
Remember when he goes to the oracle and he's he's like, should I go to war? And then like he's like, no, you're gonna lose or something like that. But turns out he was bribed by the Persians. Right. Anyways, the oracle of Delphi is supposed to tell the future. Okay. And predict the future. So this thing is predicting how the repair is going to go. Okay? Because the repair mechanisms in cells is extremely complicated. Yes. But at the end of the day, there is a hidden pattern. Okay, and that's what AI is all about.
Like we just
It's
talked
got to
about.
be it's got to be a complicated set of data, but there has to be some hidden pattern, and AI is really good at picking out those hidden patterns. The pattern could be based on the cellular environment. It could be based on like where along the genome that this cut is we need this cut to happen, or like how the chromatin, which is like the proteins that wrap the DNA around, how that stuff works, right? So all of those things are what go into this AI, and what comes out is a way to predict how the repair is going to go, where we should even do the cut so that the repair is best, right? And like minimal amount of work. And the other cool thing is they've made this kind of like glue, this repair glue. Okay. What it does is it's a custom piece of DNA that guides the cell's DNA repair mechanisms to come and do it right. So we're not just like hoping.
Yeah, it's not hoping
right
anymore.
It's like nope, it's over here. It's like a runway for the enzymes to like come in and be like, oh, okay, cool, I'm gonna do this repair over here. It's gonna be really nice. There's gonna be no more scars. That's what we call them. Like when CRISPR like makes a cut and then like some random crap happens, it's called a scar in your gene, and it's not a good thing, right? It might even kill the whole project itself.
This makes me think of the scene in Foundation. If there's any Foundation fans out there, the Apple TV show
that's
based
a great,
on the Isaac Asimov books. There's spoiler alert. The one of the people that serves the main, royal family is this like sentient robot, and anytime she gets injured, it's just the healing kind of happens in real time, and just the visual of that makes me think of this kind of this new glue, the ability to basically control
at a molecular level,
at a molecular level, which is incredible in its own right. And so we're sort of expanding the toolkit for gene editing to not just be able to, we have the map of DNA, like we know what different aspects of our DNA do, so we can be like, oh, like if we were to cut here, certain outcomes are going to happen. We then created the tool to do the cut, and now we are beginning to develop a sort of planning tool with the AI system that helps us simulate what are the outcomes going to be to a cut and how the body will respond as one aspect. But as a second aspect, we've also created some helpers to guide that process in addition to being able to sort of look at how that process is going to transpire. So it's not only a planning benefit, but there's also an execution.
There's a part of this that is execution itself at the molecular level,
which is, and so now when you can cut and heal and have some level of efficacy
in the entire top-down process,
top-down process, from identification to extraction to repair, obviously, my mind goes to the implications. You can now have more consistency in expected outcome to changes made by CRISPR.
That's exactly the point,
which obviously makes it more viable as a therapeutic tool or whatever you want to call it for any number of reasons.
Yeah. Now we can actually start like really targeting and in a safe way. Right? It wasn't safe before. CRISPR it obviously has its applications, but if we want to go into like real clinical therapies, where we're starting to like now go into trials like human trials and things like that, we needed this kind of mechanism that would control for the aftermath of that DNA cut. Right. Not just because the biology, if you just do one part, biology is a nested somebody in molecular biology once told me biology is nested for loops and if statements. Okay. And you don't know where there's a random break that's just written that'll just like crash your whole program.
Right.
And so you need to be extremely careful when you start editing this program. Right. To make sure you like don't get to that break point. Right. And so this is extremely important for that reason. It's paving the way to these targeted gene therapies. It's paving the way for clinical application. We need it to be safe, and this is definitely like on the road to that, right?
At least in the U.S. We can't speak for other massive nation states and their
Right,
perspective
of course.
on where the ethical line is
Yeah, yeah,
to start implementing this in humans.
yeah.
But here in the U.S.,
In this country,
where we make big, beautiful stuff,
yeah.
um,
Although this was in Zurich, but whatever.
we're good at borrowing. And we'll make it better. But this is I think a really important story though, because science always feels to me like, you know, you're sort of you're playing with God, right?
Mm-hmm.
Like we're starting to get to a place where we're sufficiently advanced in what we can manipulate and what we can engineer.
Yeah, it's getting insane.
We're getting into into sort of weird territory. Because again, it's the tool scales. Like once it exists and it gets to that level of efficiency and efficacy, it can be applied to a number of different use cases. This is not like just for X or Y. It's like it is a fundamental concept.
Yeah, actually, on that note, so they tested it on 3 working models. Human cell cultures. Okay. It worked. Mm-hmm. So this is in the petri dish. We grow human cells. On the tropical frog, which is a model organism. I think it's called Xenopus. Yeah, it's the Xenopus frog. It's a model organism that's used for generational studies. Yep. Okay. Yep. So they messed around with the DNA here, and then the kids, and it was passed down, and it was like, okay, everything's good there. Yep. And then, so it's called germline studies. Yes. And then it also worked on non-dividing adult mouse neurons. From the brain. Wow. Which is this thing where, like, you know, these cells don't have that as robust of a DNA repair mechanism as cells that do divide, and we'll need that on a day-to-day. And it worked in those cells as well. So now you can start targeting therapies like in neurodegenerative diseases, right?
Which would be an unbelievable.
That would be unbelievable.
It would be unbelievable.
Yeah, like this.
That's actually a really important small note. Yeah. That it does work on those non-dividing neuronal cells.
Like this sort of like guide, this scar edit that it does, where it's like guiding whatever the DNA repair because there's some obviously every cell has some. It's just not as robust. Yes. But like with this guide, even that can now do the work.
That is fascinating.
Yeah.
Really, really important story.
Came out.
This is we're looking at August 12th. Literally, I mean today.
Yeah, today.
It came out today.
An
unbelievable story that's going to have major implications. It's funny, being a biotech continues to be super fascinating to me.